Welcome to this explainer. Today we are unpacking an absolutely fascinating paper titled The Genie in the Bottle, Magnified Calcium Signaling in Dorsolateral Prefrontal Cortex. It was published in 2020 in the journal Molecular Psychiatry by Amy F.T. Arnsten, Dibdeep Dada, and Min Wong. Honestly, this study completely changes how we view human intelligence and, tragically, our biological vulnerabilities to diseases like schizophrenia and Alzheimer's. Let's get right into it.
Here's our quick roadmap. We'll cover 1. The cortical timescale hierarchy. 2. V1 versus prefrontal cortex. 3. The calcium signaling genie. 4. Stress and inverted U response. 5. Schizophrenia and genetic insults. And finally, 6. Aging and Alzheimer's vulnerability.
Section 1. The cortical timescale hierarchy. Building the mental sketchpad.
Okay, before we dive into the deep molecular biology here, we really need to introduce our main character. We're talking about the dorsolateral prefrontal cortex, or DLPFC. This is a newly evolved brain region in primates, and you can think of it essentially as your mental sketchpad. It's the exact area of your brain responsible for maintaining and manipulating information, even when there's no active sensory input. So it's literally how you hold a phone number in your head or, plan out your next chess move.
Now what's really cool here is seeing how the brain's timescale and complexity physically map out across its anatomy. As outlined in the 2020 Arnsten et al. Molecular Psychiatry study, there's a very clear hierarchy. Way at the back of the brain, you have the primary visual cortex, or V1. V1 operates on incredibly short timescales. We're talking like 65 to 75 milliseconds, because it has to rapidly and accurately decode fleeting visual events. But as we move forward to the brain to the DLPFC, that timescale dramatically increases. The DLPFC circuits can sustain persistent activity for seconds at a time to support our working memory.
Section 2. V1 versus prefrontal cortex. Decoding versus sustaining.
So, to really understand this, we need to compare them side by side. To decode those quick sensory flashes, V1 relies heavily on what are called AMPA receptors. These are extremely fast-acting glutamate receptors. But the DLPFC, it requires NMDA receptors, specifically ones with a special NR2B subunit. These NMDRs close much more slowly, and that allows a massive influx of calcium into the cell. And it turns out that calcium is strictly required to hold on to persistent thoughts over time.
And the authors actually proved this. The 2020 Arnsten study details these amazing physiological experiments conducted on behaving monkeys. They recorded the firing rates of specific neurons while applying tiny amounts of drugs directly to them. Looking at V1, the control condition is at our baseline, 100%. But when they introduced an experimental drug called CNQX, which blocks those fast AMPA receptors, V1 neuronal firing drastically dropped down to around 60%. But get this, when they applied the drug Rho, which blocks the NMDA receptors, it had virtually no effect. Firing stayed right up near 100%. So V1 definitively relies on AMPA.
But let's look at what happens in the DLPFC. It's the exact opposite. When recording the DLPFC, these are the specific neurons actively firing to keep a memory alive. The AMPA blocker, CNQX, does almost nothing. But that NMDA blocker, ROPE, it completely plummets the cell's firing rate down to nearly 40% of the control. That is a massive effect size. It absolutely proves the DLPFC's heavy reliance on NMDA receptors and that crucial calcium influx to maintain higher cognition.
Section 3. The calcium signaling genie, the feed-forward mechanism.
All right, so inside the DLPFC, specifically deep inside the dendritic spines of layer 3, we find a truly unique concentration of proteins. They create this incredibly powerful feed-forward loop between a molecule called CAMP and internal calcium stores located in an organelle called the smooth endoplasmic reticulum, or SER. This right here is the genie the paper's title refers to.
Actually, let me read you a direct quote from the 2020 Arnsten et al. Molecular Psychiatry paper that captures exactly how this works. CAMP-PKA signaling is known to increase calcium release from the SER by phosphorylating RYR and IP3R, while calcium in turn drives CAMP production, thus creating feed-forward signaling. Essentially, it's a loop that continuously magnifies calcium signaling. It is a brilliant evolutionary adaptation that literally gives us the power of abstract thought. But, like a genie out of a bottle, it's immensely powerful, and unfortunately, it requires absolutely perfect regulation to keep it from turning toxic.
Section 4. Stress and inverted U response.
too much of a good thing.
So what happens when we stress this system? Well, there is a very narrow optimal window for neurochemicals like dopamine and norepinephrine. The 2020 Arnsten study maps this out perfectly as an inverted U dose response. Basically, Goldilocks rules apply. You need exactly the right amount of these chemicals in the prefrontal cortex. Not too little, and definitely not too much.
Let's break that down. If you have insufficient release, like when you're super fatigued, there's low neuronal firing and your working memory suffers. Under optimal, everyday conditions, a moderate release of dopamine and norepinephrine perfectly maintains your network connections. But when you experience severe psychological stress, that excessive chemical release drives massive CAMP calcium signaling. The system just gets flooded with so much calcium that it actually triggers nearby potassium channels to open. The potassium rushes out, the cell hyperpolarizes, and your cognitive connections instantly weaken. You essentially lose your higher-order thinking in a panic.
Section 5. Schizophrenia and genetic insults. The Genie escapes.
Now we hit the really tragic part of this evolutionary trade-off. When we look at the structural findings from the 2020 Arnsten et al. Molecular psychiatry study, we can compare a healthy spine, which regulates calcium perfectly, like a tight cork in a bottle, to a dysregulated spine. In the dysregulated one, genetic or inflammatory insults have effectively broken the bottle, letting that calcium genie escape and run rampant into the cell.
The paper highlights that schizophrenia is deeply linked to genetic insults in specific regulatory proteins, things like mGluR3 or DISC1. When these mutate, the protective mechanisms unanchor. That leads to a massive surge of elevated cytosolic calcium. This runaway calcium actually triggers a severe inflammatory response, elevating immune proteins like complement C4a. That inflammation then dramatically weakens those crucial NMDA receptor connections we talked about earlier. Eventually, this sustained toxicity propels a literal, catastrophic physical loss of the dendritic spines in the DLPFC, which is a key physical pathology of schizophrenia.
Section 6. Aging and Alzheimer's vulnerability. Lifelong accumulation. But it's not just genetics, it's also time. Comparing a healthy network to an aged one, we can see the slow structural toll of an entire lifetime of elevated calcium signaling. In normal aging, our regulatory proteins just slowly degrade. This causes a chronic calcium leak. And over decades, that constant calcium exposure gradually drives the terrifying accumulation of pathological hyperphosphorylated tau proteins. You can physically see the tau proteins aggregating right on the spine's structural apparatus. This triggers neuroinflammation, amyloid deposition, and the devastating neurodegeneration seen in Alzheimer's. It's a cruel irony, but the very areas of the cortex that evolved last are the first to degrade.
To quote the 2020 Arnsten et al. Molecular psychiatry study directly one last time, thus, the genie we need for our remarkable cognitive abilities may make us vulnerable to cognitive disorders when we lose essential regulation. The very genie that grants our highest cognitive wishes, our working memory, our brilliant abstract thought, is the exact same thing that makes us susceptible to cognitive atrophy when that tight regulation inevitably fails.
So, I want to leave you with this final, rather profound thought to ponder. If our highest brilliance and our deepest cognitive vulnerabilities stem from the exact same molecular genie, how do we heal the aging or stressed brain without losing what makes us uniquely human? We clearly need therapies capable of gently coaxing the genie back into the bottle, preserving our magnificent mental sketchpad for as long as possible. Thanks so much for exploring this explainer with us today.